The next speaker is Jongmin Kim, who got his bachelor's from Pohang University in Korea in biology, did his graduate work at Caltech in my group, and is currently back in Korea at CBS Bioscience. So, Jongmin.
Thank you. So today I'd like to talk about my graduate project, in vitro synthetic transcript networks. And starting from the minimal cell, people are saying that you need like 150 genes. Then that's too many. And then hundreds of them are used for translation. So what if we can do with just transcription? Okay, we are cheating. We have like 2 enzymes, but that's all we are going to use, other than DNA and RNA. So we have DNA template, and we use RNA polymerase to produce RNA, and then we have RNase to degrade RNA signal. And RNA signal will be used to regulate the DNA templates. So we have a template which is in the off state, which is unusual because the promoter is not complete. But then we have a oligonucleotide that can complement this region, and then we can turn it to on state. And then we can add another strand that's complementary to this activating oligonucleotide, so that you can strip it off, and then we can go back to the off state. So how can we build a NOT gate out of this? So if we have a lot of activator, for example, then we start adding the inhibitor, and then at first you can start binding with a free activator that's floating in the solution. Then as you reach a certain amount, then you consume all the activator in the solution, and you start to stripping off from the template. So you go down steeply, and then there's nothing more to strip, and then you just stay at the low state. So you can, by changing the amount of template and the activator, you can set the threshold in this way. So it's similar to the neural network, but let's skip that. And we are going to measure with fluorescence. So the template has a fluorophore here, and then the activator has quencher here, so that when it's in the on state, you get low signal. But when you're in the off state, because the quencher is away from the fluorophore, you get high signal. Then you can measure it by fluorophores. So what kind of oscillators are in nature? I mean, maybe it's not really oscillator, but they can oscillate. So you can have a P53 and MDM2. P53 activates the production of MDM2, and MDM2 provides the degradation. Oops. This is wrong, for NF kappa B and I kappa B. So we decide to build an oscillator with this motif. So we have an activating switch, and then we have an inhibiting switch. So we have this RNA comes in, and then somehow, not somehow, there's activator and the DNA inhibitor. So at the beginning, there is an excess of DNA inhibitor, so the switch is initially off. And as you provide the RNA activator, it start to eat up the DNA inhibitor, so that actually the amount of RNA activator can control the amount of RNA inhibitor that's produced. And then the amount of RNA inhibitor, as similar to the NOT gate that I have shown earlier, controls the amount of activator that's being produced. So how does it work? So at the beginning we have no RNA, and then because there's no inhibition, we start building up the RNA activator. And then because RNA activator is there, it starts to promote the production of RNA inhibitor. And then RNA inhibitor shuts off the production of RNA activator, and then we go back to the first state. So in the experiment, we could see that we could oscillate for about 5 or 6 times in about 20 hours. So it's not that fast, but which is usual for biological or biochemical systems. But the funny thing was that when we run the gel to see what was going on, it was really funny because, it does seem to oscillate like up, down, up, down, up, down, but it was getting darker and darker over time. So if you plot it, it's like, okay, it's oscillating, but then why is it increasing so much? Because our initial threshold was here, and then it's operating at way higher range than when we started. So we decide to look at the gel more carefully, and then we see these kind of stuff. So what are these? So we expect that they are probably the waste products, because we are relying solely on the RNase H, which cannot degrade the RNA signal completely. I mean, it can degrade most of it, but then because of their binding requirements, they cannot degrade several bases on the 5' side of RNA in the RNA-DNA hybrid. So we are leaving this part, which is crucial for propagating the signal. So if you have a lot of this, which I call short inhibitor 2, then it can also try to compete for binding here rather than the real signal, which is RNA inhibitor 2. So if we have a lot of buildup of these guys, then the inhibition is not as efficient as it was at the beginning. So we could add that to the model. So it's hard to see, but so these lines are kind of built up with the waste. So if you subtract some certain fraction of the waste from your actual signal, then you get a new kind of plot which seems to oscillate around a fixed threshold. Then we can build another oscillator, but that's not so important. So, okay, so how many strands we use? So we use like 7 to 9 DNA strands, and then we use 2 enzymes, but that's a problem. So we wanted to do it with just RNA. So it would be great if you can tell me what kind of ribozyme to use. So, and what to do with the waste. The waste was a big problem. So although somehow the oscillator could overcome the interference, for a prolonged operation we want to get rid of it. So we can try to use dilution, or we can try to use chemostat, or we can try to find a better enzyme, or someone can engineer it for us. And then I could thank Eric and everyone in the lab. Okay, thank you.
Any questions for Jungmin? Yes. In the back.
With the in vitro oligo work like this, how do you dream up of having it to connect with biological systems somehow?
Yes, it is certainly something that we have to look into, like producing some effective molecules and stuff. But right now, the running condition is not too physiological. We use a lot of NTP and high concentration of salt, so we'll have to figure something out, yeah. We can start putting it into vesicles maybe.
So the next speaker is Lulu Chen. She did her bachelor's in biomedical engineering at Southeastern University in China, and her PhD in Shanghai Jiao Tong University in 2007, biochemistry and molecular biology. She's now at Caltech working with me on systems that generate interesting dynamics without any enzymes at all.
Okay. So, hello everyone. We know we are trying to build biochemical circuits, but as similar to the electronic circuits, we generally need sensors and actuators and core circuitry. So many progress have been made on sensors and actuators using DNA to get signal from the biochemical system or to control some biochemical device. So these achievements allow us to focus only on the core circuitry made of DNA, which will be easy to build a modular system. So I'm going to talk about a simple DNA gate motif, which appears to be suitable for scaling up. This is the abstraction of the gate. It is a node connected with wires, and each wire connects exactly 2 gates, and only different sides of the gates can be connected. And each gate consists of a left side and a right side, and it may connect to any number of wires on each side. And numbers on the wires and the inside the node that at the end of the wires represents relative concentrations of the DNA molecules. A network consists of a number of gate nodes and a number of wires between gate nodes. So we can design different networks to achieve different goals. So what is the function of the wires and the node? For a single gate, let's assume we have input signal, output signal, and field. Each wire corresponds to a signal DNA molecule which is free. And each node corresponds to a gate which can bind to or release a signal, the DNA molecule. So suppose it is bound to an output signal before the input arrives. Then when the input signal can trigger the release of the output and bind to the gate instead. Like this. Then the fuel molecule can set the input free in the same way. So that is to say, the input catalyzes the exchange of the fuel and the output. But in a network, the fuel output and input signal strengths can also play a different role with other gates. Suppose this is a gate 3, and the fuel strength can also be a right side signal strength of gate 2. So it is actually the wire that connects gate 2 and 3. So after being released, the output strength can also be a right side signal strength of gate 1. And the input strength can also be a left side signal strength of gate 4. So this is the way the gates are connected with each other. Let's take a single gate as an example to look at the wonderful molecules. When the output is bound to the gate, we call it a gate output complex. The output strength has a total domain in the center and 2 recognition domains on the side. And the gate base strength, this one, has a recognition domain in the center and 2 total domains on the side. The output strength is bound to the gate base strength by its right side. So at this point, the gate has its left toehold covered and its right toehold revealed. And the input molecule has the same components as the output molecule, except the sequences are different. So the basic reaction involves toehold mediated strand displacement. That means the input strand binds to the gate output complex via the toehold domain. And the branch migration within the recognition domain will lead to a state where the output strand is bound by no more than the short toehold, so it will quickly fall off. Now we have the output released and the input bound to the gate. So if we have some fuel strand, it can also bind to the gate and displace the input strand. So the input is released to react with the other gate complex. Compare the state before the reaction and after the reaction. The input is still there, but the output and the fuel actually change places. Here are the chemical reactions with rate K0, which depends on the length of the toehold. Now let's start the reaction with different relative concentration of the molecules. Suppose the gate output complex is 10x, the fuel is 10x, and the input is 1x. Well, 1x is the standard concentration. The input catalytically turns the free fuel signal to free output signal to reach equilibrium. Well, they are both 5x. Then the strand displacement will keep happening, but the concentration will stay at 5x each. So we know the equilibrium by solving these equations. We write the relative concentration of each molecule at the corresponding position in the gate abstraction, since different initial concentrations will lead to different gate behavior. Okay, we name it seesaw gate because it has 2 sides and it goes back and forth. In the experiment, we read the output signal using a reporter molecule, which produce increasing fluorescence signal with output. The experiment results show us behavior that is clearly catalytic because the input produces more output than the amount of input itself. In addition to catalysis, threshold can also be helpful for circuit function, for example, to clean up the leak. So for this input strand, we can make a threshold gate that has a longer toehold. Once the input reacts with threshold gate, it will turn to waste product and never be released again. Suppose the rate of this reaction is K0, and the rate of the reaction between input and gate output complex is K0. I'm sorry, that is K1. Okay, so K1 is much larger than K0 because the threshold gate has a longer toehold, and toehold mediated strand displacement rate grows exponentially with toehold length. So only after the threshold molecules have been used up can the remaining input react with the gate output complex. We also put the relative concentration of the threshold in the gate abstraction. it is written as a negative number because it absorbs the input signals. Here is the chemical reaction. We also did experiments to show that the threshold works in practice. Okay, so now the question is, what can we build out of the seesaw gate? For one thing, we can make feed-forward digital logic. This is a scheme of making OR gates. We use two seesaw gates in a row, set the threshold as 0.66 to make it an OR gate. This is the simulation, and a clear digital behavior is obtained. This is one of our experiments to test the OR gate. The output went to a clear on state, like this, or a clear off state, except there were some leaks. And it just needs more time for those three outputs to go to the same value. This is a scheme of making an AND gate, similar to the OR gates, except the threshold is set to 1.33. Again, the simulation gives good results. And this is the experiment of the AND gate, also shows a clear on state and off state. There are actually more than one scheme to make the OR gates and AND gates. Like this OR is made from a single seesaw gate with two thresholds. And it has even a better digital behavior. This AND gate is made from 4 seesaw gates, which has a comparable digital behavior to the previous OR gates. So far, we have been tested parts of those schemes experimentally. Okay, so we hope the seesaw gate is a universal motif for digital and analog circuits. We also hope it's easy to compile from abstract design to DNA, easy to synthesize in parallel, and robust to a variety of imperfections. Today, we don't have time to discuss all of those details, but I do want to tell a bit about how we can build a compiler. The first step is to design the hardware from the digital system function level to the logic gate level using an existing hardware description language such as VHDL. The second step is to compile the logic netlist down to the seesaw gate circuit abstraction. And the third step is to translate the seesaw abstraction to the DNA gate motif level. And the fourth step is to design sequences from the DNA molecules and then to synthesize them. So the first step is already well defined. And a netlist with AND, OR, NOT, NAND, NOR, XOR can be implemented with only OR gates and AND gates using dual real logic. The second step is also straightforward. The AND gates and OR gates can be implemented by seesaw gates as what we discussed before. So this is a demo of our compiler. This is a circuit with 5 inputs, 6 NAND gates, and 2 outputs. We translated it into the equivalent dual real circuit, and then into its implementation as a network of the seesaw gate. Then we simulated all 32 possible input combinations. The output was verified to be correct. And then the seesaw gates were compiled into DNA sequences like this. So of course we met some difficulties when we are working on this project, but we hope we can solve those problems soon. So if we really want to master the complexity of molecular systems, we need to turn this into computer science, into molecular programming. We need to learn the ideas from computer engineering, such as digital abstraction, mathematical specification, programming languages, and so on. Finally, I want to thank everyone in our lab. And thank you for your attention.
Questions for Lulu? Yeah, Eric.
I was really impressed with the compiler that you did, and you showed the sequences, and I was wondering what happened when you did the experiment.
What happened?
Yeah.
To which experiment?
When you showed the AND, NOT, OR gate that you compiled, and then you had 3 pages of
that means you
sequences.
want to really understand the scheme of the AND and OR gate? Or?
Well, it looked like you were able to get down to the sequences, and then I presume that you ordered them. No?
Yeah.
It was just too complicated to even attempt?
You mean the last page?
What is the most complex circuit that you have built so far?
Right. Yeah.
So the 4 seesaw gates to implement one of the AND schemes. Let me find the experiment.
Right. I remember that slide, and it worked. It looked like it worked really well.
This one, right?
So, have you done any experiments that haven't worked? And
That haven't worked.
right.
So far we did the experiment. This system consisted of 2 theta gates, and actually this result shows those combination of the 3 gates. So when we're trying to add the 4th gate. Actually, there are some leak problem, so we're trying to solve that.
I have a question. Very nice presentation. So if I'm correct, in the models that you showed for the 2 input gates, the OR and the AND, you were showing the axes with input concentration when you were predicting a fairly digital response.
Yeah.
But in the data that you showed, mostly it's with respect to time at one concentration of the different inputs. Is that true?
we didn't try all the combinations yet.
But have you tested concentration ranges so that you observed that type of very sharp digital response, or is it?
Yes. For like this one, actually we tested on 0.1x as 0 and 0.9x for 1.
Mm-hmm.
So we hope that if we even tested 0.2 or 0.3 as 0 or 0.7 or 0.8 as 1, it will still work. We hope that, but we haven't done the experiment yet.
So you're hoping that there's a sharp sort of a digital transition between the on and the off state.
Maybe you should go to the single node experiment.
The single
where you
node?
did measure the for the threshold gate. Your first experiment for the threshold gate.
Oh, I see. Okay, that one. I think it is this one. Yeah, it is actually sharp.
I see. Okay.
From here to here. This is off state and on state.
Okay.
So
Okay, but for the
possible.
2 input ones, you haven't yet done the full concentration range yet, but you expect it to behave like that.
Yeah.
Okay.